BRAF, from structure to therapy, a deep dive. Welcome, everyone, to our podcast, where today we will be delving into the fascinating world of BRAF, a pivotal molecule in cancer biology and therapy. Let us embark on this journey, exploring its structure, function, role in disease, and the latest in therapeutic strategies. Introduction to BRAF. BRAF is a serine threonine kinase and a member of the RAF family, which also includes ARAF and CRAF. These kinases are integral to the mitogen-activated protein kinase, or MAPK, and extracellular signal-regulated kinase, or ERK, signaling pathway. BRAF operates downstream of the ROS protein, where its primary role is to phosphorylate MEK, which then activates ERK. This cascade regulates critical cellular processes such as proliferation, differentiation, and survival. Structurally, BRAF possesses key domains, including a ROS-binding domain and a kinase domain that is essential for activating MEK. BRAF mutations in human cancer. Let us move on to mutations. Among the various alterations found in BRAF, the most common is the V600E mutation, accounting for approximately 80% of all BRAF mutations. This particular change results in constitutive activation of the kinase, making it signal independently of ROS. The V600E mutation is highly prevalent in melanoma, where it is seen in about 50% of cases, as well as in colorectal cancer, where it is present in around 10% of cases, and in hairy cell leukemia, where it occurs in virtually all patients. There are also non-V600 mutations, such as K601E and G469A, which, while generally displaying weaker kinase activity, remain oncogenic. Mechanisms driving tumor development. How do these mutations promote cancer? Primarily, they cause hyperactivation of the MAPK pathway, leading to unchecked signaling through ERK. This drives uninhibited cell proliferation and resistance to cell death. Mutant BRAF proteins can also bypass the normal feedback regulation provided by ROS. Furthermore, these mutations may upregulate the expression of programmed death ligand 1, or PD-L1, allowing cancer cells to evade the immune response by suppressing T-cell function. Therapeutic strategies targeting BRAF. Let us now discuss therapeutic interventions. Several targeted agents have been developed to inhibit mutant BRAF, particularly those harboring the V600E mutation, such as vemurafenib and dabrafenib. These agents are particularly effective as monotherapy in melanoma, though challenges arise due to the development of resistance. Resistance mechanisms include mutations in MEK and the activation of the epidermal growth factor receptor, or EGFR, especially in colorectal cancer. This has led to the adoption of combination therapies. The use of BRAF inhibitors together with MEK inhibitors, such as the combination of dabrafenib and trametinib, helps reduce feedback reactivation of the pathway and improves therapeutic efficacy, particularly in melanoma and non-small cell lung cancer. For colorectal cancer, combining BRAF inhibitors with anti-EGFR agents, like cetuximab, has shown promise. Clinical implications. To summarize the clinical landscape, the impact and management of BRAF mutations vary among different cancers. In melanoma, the V600E mutation occurs in about 50% of cases and is most effectively addressed with a combination of a BRAF inhibitor and a MEK inhibitor, resulting in improved survival rates. For colorectal cancer, approximately 10% of tumors harbor the V600E mutation. Here, the best outcomes are seen when a BRAF inhibitor is combined with an anti-EGFR therapy, as monotherapy with a BRAF inhibitor alone yields poor prognosis. In hairy cell leukemia, the V600E mutation is found in virtually all patients, and treatment with vemurafenib shows a high response rate. Papillary thyroid cancer presents the V600E mutation in around 40% of cases, and dabrafenib is used as a therapeutic option, though outcomes remain variable depending on other clinical factors. Looking forward, what does the future hold for BRAF-targeted therapy? Researchers are actively developing next-generation BRAF inhibitors that target a broader range of RAF family members and non-V600 mutations. Combinations of immunotherapy, such as anti-programmed cell death protein 1 agents with BRAF inhibitors, are under investigation. Additionally, there is interest in vertical inhibition strategies that target downstream molecules such as ERK. Summary and conclusion. BRAF acts as a major driver in various cancers through hyperactivation of the mitogen-activated protein kinase pathway, with the V600E mutation being the most clinically actionable. Targeted inhibitors have transformed outcomes, but resistance remains a significant hurdle, making combination approaches vital for future progress. Thank you for joining this in-depth discussion. Stay tuned for more episodes unpacking the science behind cancer and its therapies.